Before we get into the actual steps of mitosis and meiosis, we're going to look at the cell cycle in general and how that relates especially to cancer, which is something that touches just about everybody's life. We're going to discuss what happens during the eukaryotic cell cycle, so we're done discussing prokaryotes, bacteria, and then connect this to cancer. As a reminder, why do cells need to divide? 2 reasons. We need growth and repair. And once the cell cycle starts getting out of control, we have too much growth, and this leads to cancer eventually. But we'll look at that later on. You need to fill in the circle on your page, just like I'm going to do with mine here. This is the steps that cells go through, including division, or sometimes they just kind of hang out here, which I'll talk about in a second. Our first stage is called gap 1. It is really important that this line right here goes past the inner circle into the outer circle, and this top line does not. Please draw it exactly like mine is, because we're going to add in some labels and this will make sense later. Our first stage of the cell cycle is gap 1. This is where a cell is growing. It's doing reactions like breaking down sugars and getting energy. It's making proteins. Some organelles get copied so they can be used and whatnot. This first part is called interphase, and it's going to include more than just G1, and that's why your first line here cannot go all the way up to the end. Most cells hang out in G1, and they often cycle through this into G0, which is resting. So they go through gap 1, they hit a checkpoint, and they say, nope, not going to divide, I'm going to hang out in G0, and they rest, and then they come back to G1, see if anything needs to happen, hit their checkpoint, say, nope, not going to divide, hang out in G0, etc. Some cells, though, when they're ready to divide, will progress onto the next phase, which is still a part of interphase, but is called the S phase, or synthesis. During synthesis, DNA is copied or replicated, and all of an organism's chromosomes are doubled. So then we can have a complete set for each new cell once we're done dividing. We have gap 1, we have synthesis. The next part is gap 2, or G2. This line here, if you look where my mouse is, make sure it extends all the way down, because gap 2 ends interphase. Interphase is really long because it includes so much stuff going on. We have a cell that's growing, hanging out, making some proteins and things. It decides it's going to divide, so then it has to copy its DNA, and then it has to prepare even more to divide, make more proteins, copy more organelles. Interphase takes a long time, and again, it's divided into these 3 sections. Once a cell is ready to actually start dividing, then it goes into mitosis. Mitosis is going to have a variety of steps, so pause me and divide up your picture just like I have mine here. The first step of mitosis is called prophase, and in the coming videos we'll talk about what happens in each of these stages. You've learned about these before, so they should sound familiar. After prophase comes metaphase, then anaphase, and telophase or telophase. Either one is correct. These 4 steps are just division within the nucleus. So remember, gap 1, S, gap 2, the cell's getting ready to divide. Mitosis, inside the nucleus, the DNA condenses. It pulls apart, and a copy of each piece of DNA goes into each separate cell, which relates to this last step here called cytokinesis, where the cytoplasm divides, and we have 2 new cells that form. If you're a little confused on these steps, it's okay, because we're going to go through them in the next video. Right now, it's important for you to understand that cells go through this resting phase called interphase. Well, they're not really resting, I shouldn't say that. They're still doing things, whether it's growing or copying DNA or making more proteins and organelles, but they haven't divided yet. It's still a parent cell at this point. When a cell is ready to divide, it goes into mitosis and cytokinesis, and you end up with 2 cells from 1 parent cell. The cell cycle is regulated, so between each of these phases, we have a checkpoint. And if a cell fails the checkpoint, let's say that its DNA is damaged and it has a mutation. If it fails the checkpoint, it can actually become programmed to commit suicide, and that's called apoptosis. So if a cell fails a checkpoint, it will be programmed to die. Cancer happens when a cell that has a mutation passes a checkpoint without going through apoptosis. So DNA gets damaged, the cell cycle is not regulated, it can pass those checkpoints no matter what. This picture down here shows some things that can cause DNA damage. And if any of these things, again, causes damage that is not caught, then the cells are going to divide way too fast. They'll divide faster than usual. You'll get too many cells that all have this mutation, and you get this abnormal mass here, and that's called a tumor. These cancer cells then take up the nutrients in the space that the healthy cells should have, and that essentially kills off the healthy cells. They're losing nutrients and allows more space for the tumor to form. Some really key terms for cancer. One is benign. A benign tumor is something that is not cancer. If you have moles on your body, a mole is technically a tumor. It's a growth that's not supposed to be there. However, if it's not spreading and it's not dangerous, it is a benign tumor. A malignant tumor is something that is cancer. This is spreading, going to other parts of the body, and copying itself so much that all these new cells have this cancer that can continue to spread. In a second here, a video link is going to pop up. This video is excellent. It goes into a little bit more detail about what cancer is. It's not very long, but I highly recommend watching it because, like I mentioned earlier, just about everybody knows somebody who has had cancer. Your next step is to conference with me. There's a next page in your notes where you need to read it, answer the questions, and come prepared to discuss them. So have the questions done before you come up and talk to me.